Now I would like to invite Dr. Wajid, Associate Professor, Jiangsu University, Xinjiang, China.
Okay.
Proceed, Dr. Wajid. Thank you.
Yeah. Okay. Thank you, Ms. Miriam, for your nice brief introduction. I'm Muhammad Wajid from Jiangsu University, Xinjiang, China. Currently, I'm working as an associate professor here. I would like to talk about the biomedical applications of bacterial-derived cellulose. Probably my talk is a little bit different from the normal what the other presenter talk about the human microorganism. This is more like an environmental microorganism that I'm interested. Okay, first I would like to talk briefly about the Jiangsu University where I'm working. It's actually a national key university in China that is located in Xinjiang city in Jiangsu province. It is ranked among the top 800 universities worldwide. This university offers 97 undergraduate programs, 213 master degree programs. Wait a moment, please. Okay, wait a moment, please. Okay, is it fine? Yeah. Yeah, and then it also offers 64 PhD programs. Currently, more than 40,000 students are enrolled in this university, including more than 13,000 as postgraduate students and 3,000 international students in our university. We have a staff of about 5,700 members that include 2,700 or more than that of faculty members. That also includes 500 full professors, and the remaining are the associate professor, assistant professor, and lecturer, and so on. We offer a variety of scholarships that include mainly the Chinese government scholarship and some provincial scholarships. So students from Pakistan, from other countries, they are welcome to apply here under this scholarship for their master and PhD studies. We have an international collaboration with more than 207 universities from 54 countries. That also includes some universities from Pakistan, about 7 of them. Okay, I will briefly talk about what I have, where I have worked so far. As I mentioned, I'm currently working as associate professor in Jiangsu University since September this year. Before that, I worked as an postdoctoral researcher at Huaqiang University of Science and Technology here in China. And also I worked as a researcher in the same university for a short time. And also I worked as a research associate in Comstate University, Islamabad in Abbottabad campus. Okay, coming to my research work, actually I'm working on bacterial cellulose. That is, I mentioned it is a bacterial product that is called bacterial cellulose. This special class of bacteria that is called acetic acid bacteria, like different genera or species are included in this, which produce cellulose. This is just I mentioned in this scheme. Actually, there are this process is controlled at molecular level, involving the role of different genes, like we call it the BCS cluster, that is bacterial cellulose synthase cluster, we call it. There are 4 different classes in that: BCS-A, B, C, D. So this for phylogenetic tree, you can see the linkage of these genes, which are present in different genera. So this shows a conserved sequence of these genes in different cellulose-producing bacteria. The production of this polymer is a very complicated process inside bacteria that involve main cellulose synthesis pathway, as well as some other pathway like pentose phosphate pathway, glycolysis, gluconeogenesis, TCS cycle. So a bit of complex process that occurs inside the microbial cells and produces the cellulose that then actually comes out the cell into the medium. For example, here I have shown here the glucose after excretion from the cell, then it comes into the medium and it is accumulated in the form of membrane. Some genetic analysis show that if some specific classes, genes are disrupted, or some site-directed mutagenesis takes place. For example, in this case, the insertion of ISS insertion sequence elements into the BCS-A gene, actually it transformed the cellulose-producing bacteria into the cellulose non-producing bacteria that is a mutant of that. So also we are utilizing this bacterial cellulose synthesis machinery for the production of a variety of functional biomaterials, for example, for hybrid luminescent fibers, some hybrid conductive fibers, some DC-based block polymers. Also, we are producing some plant tissue culture approaches for the development of hybrid cellulose producing plants. This cellulose is actually produced in the form of a mat or a membrane. It can be dried in the form of a powder, but generally it is a fibrous in nature. In the form of this mat, powder, or fibrous material, it has some interesting properties. Here I have mentioned some of the intrinsic properties that are naturally present in these bacterial cellulose. For example, it is highly pure compared to the other compound cellulose. It has high water holding capacity, and it is highly biodegradable, biocompatible, it is non-toxic, and so on. But besides that, some of its properties can be modified. For example, the site adhesion properties of biocompatibility and some mechanical strength, tissue regeneration relative properties can be modified. And further, some new properties can be added, for example, antibacterial, antimicrobial, antifungal, antiviral, some biosensing properties, some electrical conductivity, and some magnetic features can be added to bacterial cellulose by various approaches that we are currently working on. Okay, coming to my research, actually, I'm using this bacterial cellulose over the last about 10 years and working in different directions, from my PhD, then postdoc, and currently as a faculty member here. So actually, my research direction with bacterial cellulose is mainly divided into 6 directions. So first, I'm developing various functional biomaterials with this bacterial cellulose. I'm developing some cell-free systems for production of this bacterial cellulose. We are carrying out some genetic engineering strategies for improved cellulose production by this bacterial-producing species, and some carrying out some 3D printing for development some advanced functional biomaterials, and carrying some biofabrication based on biomicrobes, and some surface modification strategies are applied. And then finally, we are developing some order structured hydrogels for some innovative applications. Okay, coming to the different applications that I have already worked on that. So the first, we developed some functional biomaterial with this bacterial cellulose for the diagnosis of ovarian cancer. We developed a 3D model with Thetason that actually allowed the growth of the cells not only on the surface but deep into the matrix. Thus, this kind of 3D cell model can be effectively used for the early diagnosis of ovarian cancer. We already carried out some molecular analysis with notch receptor system that shows some promising result for early diagnostic application. So further, we developed some functional material with some nanomaterial, like in this case with selenium, for the development of for the application in osteosarcoma therapy because of the anti-cancer properties of this material selenium. And also in one of my study, I co-culture some bacteria and some fungi, like in this case, Pseudomonas aeruginosa, and some fungal species to produce an anti-cancer agent that is used as anti-cancer drug that is called 5-MPCA is a short form. This drug effectively reduces the growth of some cancer cells, so we believe that this kind of drug can be effectively used for the cancer treatment. Okay, these were some cancerous application cancer diagnosis and treatment. Then moving, we have used this material for some wound dressing application and some cosmetics. For example, in one study, I developed surface modified the bacterial cellulose with through acetylation, and developed some acetylated bacterial cellulose that through some interesting morphology. The acetylated bacterial cellulose supported the growth of cells not only on the matrix but deep into the matrix, and then it was actually used for the because this was a transparent material, so this was actually used for the development of transparent wound dressing as well as some cosmetics application. For example, different cosmeceuticals can be impregnated into this cellulose. As you can see in that mask, a mask is shown that is usually used in the cosmetics for cosmetic applications. Okay, also we developed some wound dressing materials with bacterial cellulose. So for example, for skin burn, for the treatment of skin burns, in this case we use a natural compound, that curcumin, that is used in preparation of some salan, like you know. And then we applied this material for the treatment of one wound in animal model that effectively actually closed the wound surface. So that was another interesting study. Also, I developed some drug delivery system with this bacteria cellulose. In this case, we modified the cellulose with the polyaniline. Then we developed a sandwich-like structure with bacteria cellulose that actually supported the drug release in both pH environment and under electric field. So that was a controlled drug delivery system that showed a promising result and can be used for various applications. Beside that, I developed some biosensor with bacterial cellulose. In the first, for example, in one study, I modified bacterial cellulose with carbon nanotubes and polyaniline that actually, and another one that we modified with reduced graphene oxide and silver nanoparticles that show some good antibacterial properties and some conductive properties. In one another study, we modified bacterial cellulose with carbon nanotubes and then polyethylenamide, and then some bacteriophages were immobilized on the surface of this material that was actually used for the detection of specific bacteria. For example, in our case, we use Staphylococcus aureus, that is a foodborne bacteria. Okay, beside that, I use this material for the development of bone tissue materials, for example, with hydroxyapatite and some nanomaterials. Okay, the second important part of my research is the basic production through cell-free system. I effectively developed a cell-free system that was free from the living cell, but instead it was comprised of enzyme. That system effectively produced cellulose with interesting properties, and then the same system was used for the development of some antimicrobial materials. Okay, also I carried out the 3D printing with this bacterial cellulose. As you can see, we have a 3D printer in our lab that was used for the development of 3D printed scaffold. We used this extrusion printing system and optimized various parameters for the printing. For example, the viscosity of the solution used, the temperature, the speed, and so on. And a lot of materials like gelatin, arginate, small intestine, cellulose, and some others were printed successfully, which show some interesting properties. Okay, in one particular study, we carried out the 3D printing of BC bacterial cellulose with gelatin and hydroxyapatite. The scaffold was used for the bone tissue engineering application, which shows some interesting mechanical properties and biological properties. Okay, also I carried out the 3D printing of live microbial cells. The printed scaffold effectively shows some interesting properties and supported the growth of different microorganisms. For example, if we use E. coli and Saccharomyces cerevisiae. So that was another interesting study. Also, we are manipulating the living surfaces of different microorganisms and control different properties. For example, we are carrying the wet stretching, genetic manipulation, and some galvanometric control for improving the properties and the orientation of the cellulose fibers. And that study actually resulted in the development of some microstructural materials which can find application in the development of some specific tissues, for example, for muscles. Okay, in one latest study, we carried out the biofabrication of bacterial cellulose. In that case, we carried out the stretching, and as well as electric field was applied to develop the some structural material for skin tissue engineering applications. Okay, we are using different microbes, for example, viruses, algae, bacteria, and fungi, and utilizing their surface properties for the development of various nanomaterials. For example, as you can see in the graphic, I have just summarized some of them here by using yeast templates of algal templates and my bacteria and viruses. Also, I have developed some functional bacterial cellulose-based hydrogels for a variety of biomedical applications. Okay, thank you. I would like to acknowledge some of the persons who have been always there to assist my research and my achievements. Okay, thank you so much. I have some invitation for submission of manuscript in different journals there where I'm working as the associate editor. So if you have any related work with these issues, you are welcome to submit your work. Thank you.
You're welcome, Dr. Wajid. Your material was so informative, and I honestly had loved it because I have so much interest in cancer-based research. Thank you very much.
Thank you. Thank you so much.